Hello, everyone. My name is Serenade Trevino, and I work in Dr. Sue Manchu's lab at the University of Texas, Rio Grande Valley. Today, I will be presenting a study of the effects of sodium alginate concentration on the physical properties of injectable alginate scaffolds for the treatment of glioblastoma. Glioblastoma is a rare and aggressive form of brain cancer. It supplies itself through the process of angiogenesis, which is the formation of new blood vessels using pre-existing and healthy ones. The current standard of treatment is tumor resection, chemotherapy, and radiotherapy, but the high recurrence rate shows that there's still a need for an alternative treatment method. Alginate is a biocompatible polymer that can be used to fabricate injectable scaffolds to fill the leftover defect sites after tumor resection. And minocycline is commonly used as an antibiotic, but has been shown to have anti-angiogenic properties that can be useful in treating GBM. Our objective was to investigate the effects of varying sodium alginate concentrations on scaffold properties and their potential for GBM treatment. And we hypothesized that sodium alginate concentrations would have an effect on pH, wet and dry weight, gelation time, dimensions, degradation, drug release kinetics, and cell viability. The way that we fabricate these scaffolds is by dissolving our components in water first, and then we mix and homogenize to obtain a homogeneous solution. After this, we then pipette the solution into 24 well plates to obtain a uniform scaffold. And you can see what they look like after about an hour there in the pictures in the methods section. So we chose groups based on the sodium alginate concentrations of 1, 1.5, and 2% weight per volume sodium alginate. And after the fabrication, the process varies depending on which study we are conducting. The first study that we conducted is our characterization of scaffolds, and you can see the data in table 1. Here we can see that the increase in sodium alginate leads to an increase in cross-linking. And this cross-linking is the process that happens between the sodium alginate and the ion, in our case is calcium carbonate. This cross-linking forms a gel matrix, which leads to the formation of a scaffold. And so the increase in cross-linking leads to sturdier gels. And this is shown in the significantly greater diameter and height of the 1.5 and 2% groups compared to the 1% group. We can also see this in the increase in dry weights as the sodium alginate concentration increases in the groups, and also a decrease in gelation time. The decreased dilation time is because of the increased sodium alginate concentrations, and also this leads to cross-linking increase, and this is because of also an increase in GDL, which is our accelerator of the cross-linking process. pH values were not significant between the groups, but they all showed a uniform increase over the 60 minutes that we recorded the pH, and they all stabilized close to about a value of 6. From there, we evaluated the cumulative degradation rate of our scaffolds, and we did this by first submerging a very large batch of scaffolds into DPBS, and we incubated them at 37 degrees Celsius. At each time point over the course of 10 weeks, our time points were day 0, day 14, day 35, and day 70. We would take a batch of the scaffolds and then freeze them, and we repeated this process at every time point until all the scaffolds were frozen. And after they were, we lyophilized to obtain the dry weights of the scaffolds at each of the time points to form a graph as shown in figure 1. And here we can see that all of the groups demonstrated similar and steady degradation rate until after day 35. And here, after this time point, the scaffolds of the 1% group actually degraded significantly faster compared to the rest of the groups, with 1.5% degrading the slowest. And this will be investigated in further studies because this is the middle percentage. And so we don't know exactly why that was the slowest to degrade. But we also see that the decrease in sodium alginate concentration results in reduced cross-linking and a faster degradation. From there, we investigated our drug release kinetics of the scaffolds, and this process is a little similar to the degradation study process. We submerged drug-loaded scaffolds this time in, again, DPBS, but we used the same batch of scaffolds throughout the course of our 14-day study. And at each of the time points of day 1, 2, 3, 7, 10, and 14, we removed the DPBS, and this held the drug that was released from the scaffolds. And we took a sample of it and analyzed the absorbance in the spectrophotometer. and we collected it into the graphs shown in Figure 2 and Figure 3. Figure 2 shows the cumulative drug release over the course of 14 days, and Figure 3 shows the amount of drug released at each time point over the course of 14 days. What we see here is that the initial burst release shows 1% having a significantly higher burst release than the rest of the groups. And this is because of the lower cross-linking, again, in the lower percentage groups. And from day 7 to day 14, the 2% scaffolds showed significantly higher amount of release compared to the 1 and 1.5% scaffolds. And this is possibly due to, again, the increase in sodium alginate leads to an increase in cross-linking. This leads to an increase it leads to an increased tightness of the gel matrix. And this possibly leads to the higher concentration groups retaining more of the drug than the lower concentration groups, which also possibly leads to the higher burst release of the 1% group in the beginning of the study. And after this, we evaluated the effects of our scaffolds on cell viability. We did this by submerging our scaffolds, both blink and drug-loaded, into cell culture medium instead of DPBS. And we incubated the scaffolds in the cell culture medium for 24 hours before treating U87 glioblastoma cells with the blank and drug-loaded scaffold release solution. And we used unaltered cell culture medium as our control treatment in this study. After we treated the cells, we then performed an MTT assay after 48 hours, and the results of the MTT assay are shown in the cell viability graph in Figure 4. And so here we can see that the minocycline-loaded scaffolds resulted in an increased cell death compared to the control and blank scaffold release solution treatments. And there was no significant difference between the high and low percentage groups. We used 1% and 2% groups to show to see if there was a difference between a higher and lower percentage. But again, like I said, there was no significant difference between the groups. But there was a significant difference between blank and drug-loaded scaffold. Also to note is that the blank scaffolds resulted in a small percentage difference compared to the control group, and this was actually due to the inhibition of cell proliferation instead of cell death, which we saw from the drug-loaded scaffolds. This inhibition of cell proliferation will be further investigated in future studies. So overall, what we concluded about the scaffolds, our injectable alginate scaffolds, is that an increase in sodium alginate concentration leads to a sturdier gel, and this leads to an increase in height and diameter and also a decreased gelation time. Additionally, a workable gelation time and uniformity was reached for all of the concentrations that we tested. And the increase in cross-linking results in a tighter matrix, which leads to a slower degradation rate and a lower burst release of the drug. Our drug-loaded scaffolds were able to induce cell death compared to the blank scaffolds, and alginate scaffolds show to be a promising alternative therapy for GBM treatment, as they can be used as delivery vehicles for other anti-cancer drugs like minocycline, including minocycline as well. And future studies will focus on increasing the amount of drug loaded to optimize the amount of cell death these scaffolds can induce, and also investigating the inhibition of cell proliferation of our blank scaffolds. Thank you.